Today we'll focus on cortisol, a steroid hormone that sits at the intersection of metabolism, inflammation, immune function, and stress physiology. We'll start with how cortisol is made and regulated in the body, then move on to how it works at the molecular level, and finally examine how medicinal chemists have modified cortisol to create powerful therapeutic drugs, along with trade-offs that come with manipulating this pathway. Cortisol is often called the stress hormone, but that label understates its importance. Cortisol is essential for life. It maintains blood glucose during fasting, restrains inflammation, and coordinates physiological responses to stress. Both too little and too much cortisol can be dangerous, which makes this pathway an especially important and tricky drug target. Cortisol is a hormone, so let's first define what a hormone is. Hormones are chemical messengers. They are nearly always made by endocrine glands, and they travel in the blood. Their signals can affect different organs and tissues in different ways. Hormones are especially important for growth, metabolism, appetite, puberty, and sexual reproduction. Hormones are defined by what they do rather than what they are made from. So hormones aren't a type of molecule in the way that proteins or carbohydrates are. Hormones can be made from different types of molecules, including proteins, steroids, or any other type of small molecule. For a formal definition, a hormone is a biologically active signaling molecule produced by a cell or tissue that is released and travels to influence the behavior of other cells or tissues, often at very low concentrations, by interacting with specific molecular targets or receptors. To be called a hormone, a molecule has to act as a signal that affects the activity of cells, causing them to change what they're doing. It also needs to be sent out away from the cell where it was made to affect cells in other parts of the body. In humans, hormones are released directly into the bloodstream. This allows them to travel quickly around the body. Hormone molecules are released from the bloodstream. They spread out to reach all parts of the body. The cells that are able to respond to the hormones, their target cells, recognize them through the use of special receptors that only bind that one particular type of hormone. Organs that produce and release hormones are called endocrine glands, and the entire system of endocrine glands and hormones is called the endocrine system. When we think about hormones, we often imagine signals that travel through the bloodstream to distant organs. But in reality, hormones and hormone-like signals can act over multiple spatial scales, depending on how and where they are released. At the top, we see autocrine signaling, where a cell releases a signaling molecule that binds to receptors on the same cell. This type of signaling is common in immune cells and cancer cells and allows a cell to reinforce or fine-tune its own behavior. Next are the gap junctions, which represent the most direct form of cell-to-cell communication. Here, small signaling molecules move directly between adjacent cells through protein channels. While these signals don't travel far, they allow for rapid and highly coordinated responses across tissues. In paracrine signaling, a signaling cell releases molecules that diffuse over short distances to nearby target cells. Many growth factors, cytokines, and inflammatory mediators act this way, creating local signaling environments rather than whole body effects. Finally, endocrine signaling is what we traditionally associate with hormones. In this case, signaling molecules enter the bloodstream and act on distant target tissues. Cortisol is a classic example of an endocrine hormone produced in the adrenal cortex and distributed system-wide. The key takeaway is that hormones are defined not just by distance, but by receptor-mediated signaling and physiological integration. Cortisol primarily acts as an endocrine hormone, but its effects are shaped by local tissue context, receptor expression, and feedback regulation. This slide highlights a special class of signaling that sits at the interface between the nervous system and the endocrine system, known as neuroendocrine signaling. At the top, we see classic neurotransmitter signaling. An action potential travels down the neuron, triggering the release of a neurotransmitter, such as dopamine, into the synaptic cleft. The signal acts over a very short distance and affects a specific nearby target cell. This type of signaling is fast, localized, and highly precise. At the bottom, the same electrical signal leads to a very different outcome. Instead of releasing a neurotransmitter into a synapse, the neuron releases a neurohormone, such as gonadotropin-releasing hormone, GnRH, directly into the bloodstream. Once in circulation, the signal can travel throughout the body and reach distant target organs. Importantly, only cells that express appropriate receptors will respond. Cells without the receptor, as shown here, non-target cells, are effectively blind to the signal even though the hormone reaches them. Neuroendocrine signaling allows the brain to convert electrical activity into long-lasting, system-wide physiological responses. This is the mechanism that links stress perception in the brain to hormone release from peripheral organs, including the adrenal cortex and the production of cortisol. Cortisol regulation is a classic example of neuroendocrine control, where neural signals are translated into endocrine outputs that shape metabolism, immunity, and inflammation across the entire body. This slide shows the hypothalamus and the pituitary gland, which together act as a central control hub linking the nervous system to the endocrine system. The hypothalamus is a region of the brain that senses internal and external conditions, such as stress, circadian rhythm, and metabolic state, and it translates that information into hormonal signals. Just below it sits the pituitary gland, often called the master gland. The pituitary doesn't decide what to do on its own. Instead, it responds to instructions from the hypothalamus and distributes signals to peripheral endocrine organs. The pituitary has two functionally distinct regions. The anterior pituitary, or adenohypophysis, releases hormones like adrenocorticotropic hormone, ACTH, thyroid-stimulating hormone, TSH, growth hormone, and the gonadotropins. These hormones act on other endocrine glands or tissues to stimulate hormone production or physiological responses. The posterior pituitary, or neurohypophysis, is different. It does not synthesize hormones itself. Instead, it releases hormones such as oxytocin and antidiuretic hormone that are made in the hypothalamus and transported down the neurons for release into the bloodstream. For cortisol regulation, the key pathway to focus on is the hypothalamic release of CRH, or corticotropic releasing hormone, which stimulates the anterior pituitary to release adrenocorticotropic hormone, ACTH. ACTH then acts on the adrenal cortex to drive cortisol synthesis. While this diagram includes many hormones, the important concept is hierarchical control. The hypothalamus sets the signal, the pituitary amplifies it, and the peripheral glands, like the adrenal cortex, produce hormones that act throughout the body and feed back to regulate the system. In the next slide, we'll focus specifically on the hypothalamic pituitary adrenal axis and follow the cortisol signal step by step. This slide brings together the two major biological pathways that make up the stress response system. While they are activated by the same stressor, they operate on different time scales and use different signaling strategies. On the left is the sympathetic adrenal medullary axis. In response to an acute stressor, the hypothalamus activates the sympathetic nervous system, and signals travel rapidly down the sympathetic trunk to the adrenal medulla. This triggers immediate release of catecholamines, primarily epinephrine and norepinephrine. Note that epinephrine is also called adrenaline. This pathway is fast, neural, and short-lived. It prepares the body for immediate action by increasing heart rate, blood pressure, and glucose availability. This is the classic fight-or-flight response. On the right is the hypothalamic pituitary adrenal axis, or HPA axis, which is slower but longer lasting. Here, the hypothalamus releases corticotropin-releasing hormone, or CRH, which stimulates the pituitary gland to secrete adrenocorticotropic hormone, or ACTH. ACTH then acts on the adrenal cortex, leading to the synthesis and release of the glucocorticoids, most notably cortisol. So cortisol is a glucocorticoid steroid. Unlike catecholamines, cortisol acts through intracellular receptors and alters gene expression. This makes its effects slower to appear, but much more sustained. Cortisol helps mobilize energy stores, modulate immune responses, and maintain homeostasis during prolonged or repeated stress. The idea is that both pathways are activated by stress, but they serve different physiological roles. The sympathetic adrenal medullary axis handles immediate survival, while the HPA axis manages longer-term adaptations. Most cortisol-based drugs target this second pathway. This slide shows the HPA axis, which is the primary endocrine pathway responsible for regulating cortisol release. Step 1 begins with stress perception. A physical or psychological stressor is produced by the brain and integrated by regions such as the hypothalamus and the hippocampus. Step 2, the hypothalamus responds by releasing corticotropin-releasing hormone, or CRH, into the hypophyseal portal system. This is a local vascular connection between the hypothalamus and the anterior pituitary. Step 3, the anterior pituitary secretes adrenocorticotropic hormone, or ACTH, in response to the CRH stimulation into the systemic circulation. Step 4, ACTH travels through the bloodstream to the adrenal glands, where it acts on the adrenal cortex to stimulate the synthesis and release of glucocorticoid hormones, primarily cortisol. Step 5, circulating glucocorticoids act on target tissues throughout the body. They increase glucose availability, alter metabolism, and suppress immune and inflammatory responses, key adaptations during prolonged stress. And step 6, cortisol also feeds back to the brain. By activating glucocorticoid receptors in the hypothalamus, cortisol provides negative feedback that reduces further CRH and ACTH release. This feedback loop ensures that cortisol levels rise when needed, but do not remain elevated indefinitely. Because cortisol regulates gene expression through intracellular receptors, drugs that target this pathway can have powerful therapeutic effects and equally powerful side effects. This slide introduces the glucocorticoids as a class of steroid hormones produced by the adrenal cortex, with cortisol as the primary endogenous example. One of the defining features of glucocorticoids is that they are pleiotropic, meaning that a single molecule produces multiple distinct effects across different tissues. Cortisol plays a central role in the stress response, but its actions extend far beyond stress alone. It regulates metabolism by promoting glucose availability, influences blood sugar levels, and reshapes how cells use energy. At the same time, cortisol suppresses inflammation and modulates immune activity, helping prevent excessive or damaging immune responses. These diverse effects occur because glucocorticoid receptors are widely expressed throughout the body. When cortisol is released into the circulation, it does not act on just one target organ. It coordinates responses across many tissues simultaneously. This pleiotropy is what makes cortisol essential for survival, but it is also what makes glucocorticoid drugs challenging. When we use cortisol-based drugs therapeutically, we rarely affect just one pathway. Desired anti-inflammatory effects are often inseparable from metabolic and endocrine side effects. To understand why cortisol has such wide-ranging actions and how medicinal chemists try to control them, we now need to look at the biochemistry of cortisol, starting with its structure and receptor interactions. In the next section, we'll zoom in from physiology to molecular detail and examine how cortisol's chemistry enables these system-wide effects.